Hey everybody, so we're here to talk about the process of protein synthesis, and we're focusing on transcription and RNA processing in this first part. And this little model here is kind of like the rules. So we go from DNA to RNA, that's our transcription. We're rewriting the DNA in the form of RNA. And then we go from RNA to protein, that's the translation. We're going from the language of nucleic acids to the language of amino acids. So transcription will be the synthesis of RNA using information from DNA. So we're going from what's on the DNA to an RNA strand. It allows for the message to be transcribed and leave the nucleus. DNA doesn't leave the nucleus, but RNA can. Translation, which happens outside of the nucleus, is synthesis of polypeptides using the information from RNA. So in the nucleus, we do transcription, DNA to RNA. RNA can leave the nucleus and go to a ribosome, which is where this happens. And that nucleotide sequence, that RNA, is then read and translated into an amino acid sequence. So to do this, we need to make sure we understand the key types of RNA. Understand that there are actually more types of RNA than the main ones we always talk about. We're just focusing on the big ones. So as we go through transcription and translation, transcription in the nucleus, translation at the ribosome, there will be three key RNA molecules that we deal with. The first will be messenger RNA or mRNA. Then we will have ribosomal RNA, our RNA, and transfer RNA, which is our tRNA. So starting with the messenger RNA, you'll notice the messenger RNA, we use the DNA as a template, open it up, and build our RNA transcript, our RNA strand. So it carries the message, and this is allowed to leave the nucleus. So mRNA carries the information from the DNA to the ribosome, which is in the cytoplasm. Our RNA leaves the nucleus, DNA doesn't. So we copy it and carry it out. Ribosomal RNA helps form the ribosome. So part of the actual ribosome structure that we're going to be using to make proteins is built out of RNA, specifically ribosomal RNA. It will help link amino acids together when we're making the proteins. Transfer RNA is all about transferring, translating from one to another. So these are what makes the process of translation possible. We have a RNA section, that's our anticodon loop down at the bottom, and we have the end that actually connects to the amino acids. So this is where we make those connections between RNA and amino acids. So each tRNA can carry a specific amino acid. So this specific tRNA is coding for one specific amino acid. It then can attach to the mRNA via the anticodon loop. So this end is all about complementing the messenger RNA. So if we have a C on the messenger RNA, it would pair with a G on the tRNA. A U would pair with an A, so forth and so on. So it's always complementary to the mRNA. Anticodon complements the codon. and complementary, always basically it's what pairs with what pairs with. So this allows information to be translated from RNA to a peptide sequence, amino acid sequence. So when we're thinking about the genetic code, we know that DNA contains sequences of nucleotides that code for specific proteins. Okay. We read these sequences in groups of 3 that we call triplets. Specifically, in DNA, we typically refer to it as a triplet code. During transcription, one DNA strand will actually be transcribed. It is the template strand. The other one is the opposite strand. So the template strand may also be known as the non-coding strand, minus strand, antisense strand. Okay, and so what happens here? The mRNA molecules formed are anti-parallel. Remember, every time we make a copy, we're always copying in the opposite direction. And complementary, so opposite, A pairs with U. Remember, U in RNA to the DNA nucleotides. So A pairs with U, C pairs with G. mRNA nucleotide triplets. So DNA, we typically refer to them as triplets. In mRNA, we will refer to them as codons because they have the code on them. Codons will actually code. They tell us what amino acid we're talking about. And so we have a chart that we put the mRNA codon into it, and it tells us what amino acid goes with that. Here is one example of that chart. So we have 64 different codon combinations. Taking the A, C, U, and G for the first letter, second letter, and third letter gives us 64 different combinations. But there's actually only 21 amino acids. So 61 codes for amino acids. 3 of the 64 are what we call stop codons, so they basically say stop building. These are universal to all life as we know it. So every living thing we have here on Earth that we're aware of at this time use this specific system. There's 64 codons, but there's only 20 amino acids plus the stop. So this allows for redundancy. We may have more than one codon that's going to code for a specific amino acid. And we always function in what we call the reading frame. Remember, we said codons are sets of 3, 3, 3, 3. When we're talking about protein synthesis, it's all about the 3. 3, 3, 3. So the codons have to be read in the same groupings. If we shift over one letter, that changes the reading frame, which means we're going to totally get different proteins. So each set of 3 has to be read correctly. So if we say, the fat cat ate the rat, that makes perfect sense. But if we shift that frame, even by one single letter, we get something different. Hef, ak, ata, tet, her, at. Notice the last one isn't even 3 letters. So all it takes is one little shift in that set of 3, that reading frame, and it doesn't work anymore. It doesn't make any sense. So those reading frames are really important. And when we start talking about mutations, we're going to see that reading frame difference. Okay, quick check. If the DNA template strand reads 3' to 5', A, C, G, A, G, A, what's the messenger RNA transcript going to read? So A pairs with U. Remember, RNA has U, not T. C pairs with G. So we're going to get UGCU. And notice that the 3' and the 5' are also opposite or anti-parallel. Using the mRNA transcript from number 1, okay, so this red that we just created, what amino acids will be produced during translation? So we're going to use that codon chart for this. And our first codon is UGC, and our second codon is UCU. Remember, reading frame's important. So we get cysteine and serine. So that's kind of an overview of the process. Now let's look specifically at transcription. Remember, transcription's in the nucleus from DNA to messenger RNA. So there are 3 key steps involved in this process. Initiation, where we're going to start it. Elongation, where we go all the way till we get to the end of the gene, and then termination, when we have to stop. So starting with initiation, transcription begins when RNA polymerase molecules attach to a promoter region of the DNA. So if you think back to when we talked about replication, we had a DNA polymerase that attached to a primer and went along. So here we have an RNA polymerase that's going to attach to a promoter, a region in front of the gene of the DNA. We don't need a primer in this case, like we did with the RNA replication. The promoter regions are upstream, so they're up ahead of the gene. So as long as we attach to the right promoter region, we will transcribe the whole gene following it. So here, notice we have our, what we call it, the TATA box, the promoter region. We look for the TATA box, TATA, in eukaryotes. And transcription factors will help that RNA polymerase bind right there to the TATA box, the promoter region. In prokaryotes, the RNA polymerase will bind directly onto that promoter region. We don't have to have these specific transcription factors that eukaryotes need. So basically, eukaryotes need an extra little layer of glue. In prokaryotes, it just sticks right on. Notice the promoter is always upstream of the gene, which we're showing in green here, that we want to transcribe. The point being that we want to make sure when we're making that messenger RNA, we get the whole gene sequence we're looking for. Step 2 is elongation. So once the RNA polymerase has attached and opens, it will open up the DNA and read those sets of 3, those triplets, on the template strand to build the messenger RNA. And so we're moving 3' to 5' direction and building that messenger RNA. And remember, we're always building in the opposite direction, so we're elongating 5 to 3. And we're just making it longer and longer as we're reading through all of the genes that we want to transcribe. And then we get to, so the RNA polymerase moves downstream. It opens little sections at a time, so it's not big, wide open, it's just sections. It's going to pair RNA nucleotides to that DNA template. And as that RNA gets longer, it's going to kind of bend down, peeling away from the DNA, so the double helix can reform. So a single gene can be transcribed simultaneously by 7 RNA polymerases because they're each at a different location, and we're only opening and then reclosing small sections at a time, so they can be running basically one after another after another. That way we can make more messenger RNA to ensure that we're making enough of whatever product we need. And then ultimately we reach the end of the gene that we're trying to transcribe. So in prokaryotes, transcription proceeds through a termination sequence. There's some sort of sequence specifically on that gene that causes it to terminate. We get a termination signal. The RNA polymerase will detach. Messenger RNA transcript is released, and it's going to head off to be translated. Doesn't need any modifications. We don't have to do anything but send it off to translate. For eukaryotes, however, RNA polymerase transcribes a sequence of DNA that we call a polyadenylation signal sequence. This codes for a polyadenylation signal, AAU, AAAA. Okay. This releases what we call a pre-mRNA from the DNA because the mRNA still has to be processed for eukaryotes. It has to be modified before translated. So for those eukaryotes, we're going to move into what we call pre-mRNA modifications. There are 3 modifications that will occur before it's actually ready to translate. We're going to put on a 5' cap. We're going to add a poly A tail, and we're going to do a little RNA splicing. We're going to cut some pieces out and then put the rest of it back. Okay, so to add the cap, on the 5' end, we want to put on a little modified guanine nucleotide cap. This kind of helps protect the beginning, the start codon part, and helps it be recognized. And then at the end, we're going to put on a poly A tail that has a whole lot of little A's because this will also signal this is the end, we finished. Both of these help the mature mRNA leave the nucleus without damaging the gene we need. It helps protect from degradation. And it will also help the ribosome actually attach to that 5' end, and so that we can then use it to build the protein. So it's really important to add these 2 pieces to the end. And then the last part, that RNA splicing. So we have parts that are called introns and part that are called exons. I always remember that introns stay in the nucleus. Introns in the nucleus, and exons will exit the nucleus. So we're going to take the introns, we're going to cut them out, we're going to splice or join the exons together, and then with the cap and the tail, we are ready to ship it out. So introns are little intervening sequences. They don't code for amino acids, at least in terms of the gene we're trying to do. Exons would be the express sections, the sections that we need to build our protein. So introns will stay in the nucleus, exons will exit the nucleus. So why does this occur? What we found is that actually a single gene can actually code for more than one kind of polypeptide based on what are the exons versus the introns. So what might be an exon in one might be an intron in another. And so based on how we cut and splice these different coding pieces together form different proteins. So that same sequence, when we cut and paste, we can build different things out of it. We consider this alternative splicing. So once all of those modifications, we've got the cap, we've got the tail, we've done our introns and exons, and we've spliced, then the pre-mRNA is now mature mRNA and can leave the nucleus and go out into the cytoplasm to find a ribosome and be translated. Okay, so discuss the overall big picture of transcription. Transcription writes down the information from the DNA and allows it to leave the nucleus to be translated into a protein. How does initiation differ between eukaryotes and prokaryotes? So remember, in eukaryotes, we had to have those transcription factors, that extra layer of glue to attach the RNA polymerase. With prokaryotes, RNA polymerase would stick right to the DNA.